Hello, everyone, and welcome back to Psychedelic Vantage. Today, I have the pleasure of speaking with Dr. Dan Karlin, Chief Medical Officer of MindMed. Along with holding the title of CMO at MindMed, Dr. Karlin is board certified in psychiatry, addiction medicine, and clinical informatics. He is a fellow of the APA and the American Society of Addiction Medicine and an assistant professor of psychiatry at Tufts University School of Medicine. Dr. Karlin, thank you so much for coming on the channel today. How are you doing?
It's a pleasure to be here. I'm doing really well. Because exciting things happening in the company, in the field, lots to talk about.
Oh, definitely a lot to talk about. So before we dive in here, can you share with the viewers, the listeners, a little bit about MindMed and how the company plans to help those suffering with brain health disorders?
Yeah, so MindMed is a dedicated brain health disorder focused biotechnology company. And clearly, because we're on this show together, we have some adjacencies and some work in the psychedelic space, which won't be lost on anyone who's watching or listening to us. Our lead drug candidate is a pharmaceutically optimized form of LSD that in the body behaves like LSD. So we've manufactured it to pharmaceutical standards, and we've got a really neat formulation that we've used, an oral dissolving tablet formulation. But the mechanism of action is that of LSD, and we're developing that for GAD. We've got some really compelling phase 2 data in GAD, and we've recently announced that we are also starting a pivotal program in MDD. So we will be running. We've announced the designs of 3 phase 3 studies to start this year and early next year to cover both GAD and MDD.
Yeah, that's very exciting, and of course, we'll speak about the phase 2B results that your company saw with MM120. The results, obviously, because of them breakthrough therapy designation, which was it's a big deal. So Dr. Karlin, I apologize in advance here for the next question because I'm sure you have been asked this a million times already. With that said, can you share with the viewers how the research you are doing is different than the research that was done by Lykos Therapeutics with MDMA assisted therapy?
Yeah, I'm happy to discuss the topic because I think it's really important, and it's important particularly to zoom into the details of what happened both with Lykos, which they obviously have to speak for themselves and to present themselves to the world, and they currently know what FDA said to them in their CRL, and we don't. So my comments probably will be a little more limited to what we are doing and
Okay.
how that addresses some of the issues that were raised publicly at the advisory committee meeting that was held with FDA. I'll start by saying one of the upshots of the way we conducted our phase 2B is that we anticipated a lot of what was subsequently both released in draft FDA guidance and ended up being a topic of conversation at the AdCom, and likely has to do with what Lykos got in their CRL. So from the outset, we were considering how to develop MM120, which is the LSD drug that I mentioned, and our lead candidate, how to develop MM120 in such a way that it avoided potential methodological and regulatory pitfalls, which ultimately Lykos ran into. I think the first major difference is the drug. And it's impossible to talk about these things without talking about specific drugs. The modern era of psychedelic medical clinical research does tend to flatten the complexity and lump a whole bunch of different drugs into the designation psychedelic. And even FDA in their draft guidance sort of did the same thing. So at some level, fighting against that flattening, that simplification may be an uphill battle. So we end up just saying, cool, let's talk about the drugs themselves. MDMA, as Lykos is developing it, is a psychotherapy enhancer. So nobody made the claim, Lykos, really anywhere that I've seen, that somebody taking MDMA and being in a room for a duration of a session without any other intervention helps, right? It's not It doesn't behave as a psychiatric medicine that induces long-term change on its own. That creates a few really significant challenges. One of which is now you need two different interventions being applied to the same participant at the same time. Right? This is tricky. The idea of a placebo control condition also becomes more difficult because while we acknowledge, I'm sure at some point today we'll talk about functional unblinding and what that means and how we think of it, because that's one of the hot buttons in the field, and it should be talked about. Patient unblinding, which is how we usually think of functional unblinding, can mediate drug response. And we know this, placebo effect has been studied an awful lot. We understand that the patient feeling the drug or not feeling the drug may make a difference or not. Now, we think in our phase 2b research for MM120, we've definitively shown that the drug effect is caused by drug effect, not caused by functional unblinding, but we'll get to that in a little bit. When you've got psychotherapy alongside a drug intervention in a study, you have to somehow account for the fact that the other intervention being provided by the psychotherapist, is now being provided by an unblinded therapist, right? So if you take a psychotherapist who believes themselves to be an effective MDMA therapist, and you put them in a room with a participant, they also now will know the participant's drug status, whether they got allocated to drug or not, pretty reliably, in fact, very highly reliably, right? Because they're actually doing a dyadic engagement with the patient, where the patient's behavior, their descriptions of what they're feeling, their reflections on their internal state, unblind the therapist. So now you have a therapist who believes themselves to be more effective when treating someone who's received active drug than when not. And we know from the history of psychotherapy research that psychotherapists' belief in their own efficacy affects their efficacy. So you've got a double unblinding problem that's pretty hard to solve for. The MDMA effort involved multiple therapy sessions. It involved extensive preparation, extensive what they call integration. We can talk about what these words have meant and have come to mean over time. But in our development paradigm, we, from the outset, were really intentional about separating out any sort of secondary intervention from the trial. So we have required monitoring, right? This is in the guidance from FDA, and FDA has been insistent on this, that you need to have session monitors in the room. We think having a session monitor in the room is a good idea. It's not that we disagree with that. Now, the question of two, why two and how does two help becomes another question down the road. But our session monitors are very explicitly trained and instructed to not provide a psychotherapeutic intervention. So rather than doing prep sessions focused on how the drug experience might affect the person, might change them, might lead to lasting improvement in symptoms, our pre-session or pre-dose session that the participant meets the dosing session monitors and engages with them is about what's going to happen in the session. It's about understanding what someone may or may not feel in the session, what options they have for what they can do during the session, what they can do if they're feeling uncomfortable or uneasy, and included in that education is what the dosing session monitor can and cannot do. And so it's very explicit with the DSMs, it's very explicit with the participants that there is not a psychotherapeutic intervention that's happening in the room. And so what our DSMs largely do during the course of a session is sit quietly on the other side of the room and allow the participant to have their own experience. Right. The other role that the DSMs can play is getting the person some food if they're hungry, helping them walk to the bathroom if they need that, reminding them that the music is available, reminding them that their eye shades. The closest, the one part of this that people have noticed, and obviously there's always questions of what constitutes psychotherapy, and we try to be as transparent as we possibly can about this. The closest that a DSM gets to anything that even remotely is similar to psychotherapy is that if someone is having a hard time in the room, which does happen, it happens less in our phase 2, it happened less than we expected it to, quite frankly. And so we've learned a lot about what to expect from these things. But if someone is feeling anxious or concerned about the way their brain is working while they're under the influence of drugs, the DSM is able to say, remind the person about the pre-study education. Remember, we talked about that this might feel this way. We talked about that this would be the time course that this might happen. We talked about the fact that it does resolve, and these feelings you're having will go away. And we talked about the fact that you're here to focus on yourself. And that's usually enough to get someone to be in a place where they can go back and do the thing that they ultimately joined the trial to do. The DSMs also have the challenge of having to sit in a room for a number of hours. In the case of phase 3, it'll be an 8-hour monitoring period to sit in the room for 8 hours with someone who got placebo. Now, nobody knows that for sure, right? Everyone's still blinded technically, and the DSMs are blinded too. But, if someone's sitting there and saying, I'm not really feeling anything, that can be a frustrating experience for people. So the DSM also has to be able to sit there and remind them that they can read or color or draw. There are a number of activities people can do in the room if they're not focused internally. But what we saw really interestingly in phase 2 was that despite the fact that there is this fair amount of functional unblinding among participants, that we still saw a very robust placebo effect. And so the placebo effect we observed in our phase 2 was even larger than you usually see in like a traditional like SRI GAD study, where what we know is that we had a strong placebo effect despite the existence of functional unblinding, and the drug effect exceeded that very strong placebo effect. And it exceeded it by a lot. So strong degree of clinical and statistical significance. So that ultimately is more information that tells us that what we observe as a drug effect isn't mediated by unblinding.
I see. Dr. Karlin, thank you so much for that really detailed overview. I guess speaking about functional unblinding, I understand, my understanding at least is that due to the nature of psychedelics at high doses, it's important to do a dose ranging study to show a dose response. I believe MindMed's phase 2b trial in generalized anxiety disorder was the most comprehensive dose response study in the space. Can you speak a little bit about that and why it's important to be able to show that dose response in your trials?
Yeah, absolutely. We agree. We think that we did the most comprehensive dose response, dose range finding study in the field, and certainly on LSD. What we get to see through dose range finding is, as I observed already, we get to see drug response and determine what it's mediated by, determine the extent to which it's mediated by people's awareness of having got a drug. Now, functional unblinding is a reality in all psychiatric drug development, right? We're talking about drugs that make you feel something, so the fact that people feel something shouldn't be surprising. Now, as new eyes have turned to psychiatric drug development because of the psychedelic interest that's out there in the world, this is something people are encountering newly, and are right to ask questions, because trying to wrap your head around placebo effects, expectancy, unblinding, all of this is complicated. And we're the first to say that this stuff is worth talking about, is worth asking questions about. Through dose range finding, what we saw was that because we ask people about their belief as to whether they got drug, we start to know that while yes, people who do get any dose of drug reliably can assess that they did get a dose of drug, the 25 and 50 microgram groups did not perform placebo. And in fact, the 50 microgram dose group numerically performed the worst. Those people were distinctly having drug experience that they were aware of, that they said they knew they were having, but that they didn't actually get better from their GAD. And what we saw is that at 100 and then at 200, we saw very strong efficacy against GAD. About half of the patients were in remission at 12 weeks later. And so with that kind of strong response at 100 and 200, that lets us confidently move forward with the 100 microgram dose group into phase 3, because we know that it clinically performs as well as 200. So you don't get any more clinical efficacy going above 100. But what you do get is a little bit higher side effect burden during the course of the session, and you get a slightly longer session with the higher dose. So by doing this study, we could confidently land on 100 micrograms and say, look, we get the maximum clinical benefit here, and we mitigate we mitigate the risk of having adverse events during the course of the session. A really neat thing about having this one dose paradigm is that, unlike traditional psychiatric drugs, you think SRIs and others, where there's a side effect burden that people have to bear for the duration of taking the drug. In our case, while there is a very distinctive AE profile when someone takes the drug, right? The session has a distinctive, there's no question about it, that there's a distinct experience. After that day, there's really no adverse event burden moving forward, right? That you see the same rate of post-dosing day adverse events as you see in placebo. And because anytime you're collecting adverse events on a group of people, of course, you capture headaches and nausea and all the experiences of daily life that aren't perfect, right? We all have, if you were in a drug study right now, you could probably come up with something you were experiencing that would be counted as an adverse event. But that's why we have drug versus placebo studies, so we can look at the background rate of these things in the drug group versus in the placebo group and say, okay, we're seeing the same rate of, say, headache in the group who got drug as the group who didn't, so that does not seem to be exacerbated by drug. So when we do drug versus placebo, it's not just for efficacy, but it's also to define the safety profile of the drug.
I see. Again, thank you so much for the great answer there. So, MM120 was granted FDA breakthrough therapy designation for the treatment of generalized anxiety disorder. Can you explain to the viewers the significance of this, and also share about the results that were seen in the phase 2b trial?
Yeah, so we were able to bring FDA these phase 2 data where we showed at 100 and 200 micrograms, we had clinical and statistical benefit over placebo. And we're talking about a clinical benefit that's 2 to 3 times what you generally would see with the existing standard of care. So we're seeing this remarkable efficacy. The efficacy starts as soon as day 1. So 24 hours after dosing, there are some measures you can do that soon. We use the Clinical Global Impression Scale, and that scale can be done as soon as day 1. And we see a significant improvement starting as soon as the day after dosing. When we were able to start to measure the Hamilton Anxiety Scale, or the HAM-A, which is our primary measure, we can do that as soon as week 1 after dosing, because it's a 1-week look back tool. And so starting at week 1, we see that clinical and statistical benefit over placebo, and that benefit persists all the way out to week 4, or to week 12, through week 4 to week 12. And really, I think the number that strikes most folks like as the strongest bit of evidence here is we're starting with patients who are on average well into the severe category of anxiety. So the Hamilton Anxiety Scale, we had a cutoff of 20, but we had a average entry score of 30, right? And so when you look at people with that severity of anxiety and starting at week 4, persisting out to week 12, you see about 50% of folks in remission. That's pretty amazing. And I mean, when I first looked at the data, that was what struck me the most. Wow, this is really different than what we currently have available to treat patients. We brought those data to FDA and got breakthrough designation. And breakthrough designation is an indication from the agency that they think that the evidence for a drug suggests that it represents a marked difference from existing available drugs. And it's a promise, in essence, from the agency to work more closely and quickly with the sponsor to advance that drug through development. So it gives you more opportunities to meet with the agency, to get alignment with the agency, and tightens up timelines for responses. That breakthrough designation allowed us then to go into our end of phase 2 meeting, which you can have those with breakthrough or without, but we went into our end of phase 2, which happened to be not long after the Lykos AdCom. So we got to see FDA after that meeting and have a conversation about our phase 3 plans and come out of that end of phase 2 meeting with really strong alignment on what we need to do moving forward into pivotal trials.
I see. And yeah, I mean, those results were, I was really taken back when I saw the results from this trial. Correct me if I'm wrong, in the 100 microgram group, at the 12-week endpoint, I believe it was 48% remission, 65% response rate. With that said, can you explain to the viewers how is clinical remission and response defined?
Yeah, great question. So I talked about the Hamilton Anxiety Scale before, the HAM-A. That's the scale that is the regulatory accepted measure of anxiety for effectively any anxiolytic that you're trying to develop. Anxiolytic just means drug that treats anxiety. And so on the HAM-A, a score of 7 or lower is rated as a category of no clinically significant anxiety. So if someone gets below that score at the time of measurement, either week 4, week 12, week 8, whatever, then they're considered to be in remission. Response is, so that's an absolute cutoff, which means it's pretty sensitive. Like when you do percent remission, you end up in a place that's pretty sensitive to change right around the threshold, because if someone goes from a 5 to a 9, they might not feel all that much worse. They might not even notice that they feel different. It just got picked up on the scale, but now they've come out of the remission numbers. So that number exists. It's a hard cutoff. It's independent of how sick someone was when they started, but it's definitionally effective. They don't have, like, if they went in to see the psychiatrist and that was their presentation, they wouldn't be diagnosed with GAD, right? Response is a different way of looking at these same measures. So it's still the HAM-A, but instead of it being an absolute number, like a hard cutoff, it's a percentage change. And it means that people have had 50% or more decrease in their symptoms. So someone could say, let's say the average patient comes in with a 30, they drop to 13. So really substantial improvement. They're not in remission, but they're now in the mild category, right? And so that counts as a response. You can also, with a starting point where we were, there's a pretty narrow window between response and remission. So the fact that those numbers are as different as they are tells you, gives you a sense of the fact that, a lot of people sustained meaningful clinical benefit. And just to reiterate, this is from a single dose of drug with no psychotherapy being provided in the trial.
Extremely promising. So I would also like to ask you, Dr. Karlin, how many pharmacological treatments or therapies did people in the study try previously, on average, prior to being enrolled in the study?
Yeah, so we didn't have, this is an important point, which is that some folks in the space are going for treatment-resistant indication on their label. We have opted to go both in GAD and MDD to go for a broader label, right? And what a drug label allows us to do is to promote based on those label claims. We think we've every reason to believe, based on the participants we enrolled in phase 2 and who will continue to see in phase 3, that we have strong efficacy for people who have been failed by, really, I hate to say people fail drugs, it's the drugs that fail, for people who've been failed by their previous drugs, in part because treatment resistant in our current world really mostly means SRI resistant, right? So there's, yes, we have tricyclics and MAOIs, there are some atypical antidepressants, but largely when someone gets that treatment resistant label, it's because they failed 2 SRIs. And because the mechanism of our drug is so entirely different from SRIs, treatment resistance it's not really a crossover relevant thing to our drug. So what we've opted to do, instead of having people have mandatory failures on previous drugs, we take anyone who qualifies for the study. If they're on drug currently and sick enough to qualify for the study, then we carefully take, the site students, the site clinicians, but they get tapered carefully off of that drug and washed out so that they're truly getting monotherapy. And if they describe previous drug failures, they say, oh yeah, when I was this age, I took this, or this age, I took that. We document it so that we know that the most important thing about what we're capturing in the study is that we have a representative population, right? And so across the board, we try not to excessively narrow the population, because by doing so, we run the risk that our research doesn't generalize as well to everybody.
Okay, I see. So would it be fair to say that severity, it was based on someone's severity of generalized anxiety disorder that made it to be in the trial?
That's right.
So, coming back, I guess, to speaking about, what it means for someone to be in remission from these treatments and what we've seen in clinical trials so far with MM120, when I look at data released from more traditional drug development companies in the neuropsychiatry space, I personally never see the word remission anywhere. With that said, do you believe MM120 has the potential to be, what's referred to as a disease modifying treatment for certain brain health disorders?
It's a great question. And let me even zoom out a little bit to answer it, because the answer is yes, I think so. I think it has that potential. I don't think we've gathered enough evidence in our studies to conclusively say that, because I don't know that most people would think that 12 weeks of change is fully disease modifying. That seems like, that's prolonged. Being in remission for 12 weeks is really significant, and it's really important. I'm really glad we showed that we could do that. Some academic research using LSD has shown 2 treatments leading to 2 years of efficacy in a substantial part of their population. But if we zoom out a little bit, you started by saying you don't really see remission rates described in other psychiatry research.
Yeah.
That has more to do with the drugs than the desires of the researchers. I think just about everybody would love to be able to describe a remission rate from their drug.
Exactly.
But if your drug's not putting substantially more people into remission than placebo in your studies, you probably don't want to highlight that. And so response rate is used more traditionally, because you do generate response rates from, a 50% reduction is good. And you certainly see that in existing psychiatric drugs. But from the bigger picture, we live in a world in psychiatry and psychiatry research that's been defined to a large extent by the drugs we've got available and by the efficacy of those drugs. And so when we talk about drugs that for most people aren't putting them in remission and have side effects that folks don't like. And again, I always want to be careful, because it's not that these drugs haven't helped people, and they haven't helped a lot of people. And if people are sustaining a benefit from taking a drug, that's awesome. Like,
yeah.
We hope people all get treatment. We hope people are able to access treatment. We hope that treatment is paid for by payers, and that people get the thing that can help them the most that's available today. So when we compare ourselves and what we see from MM120 to what you might see in current standard of care psychiatric treatment, it's not to say people shouldn't be getting the best care they can today. Right? What's available is what's available. But when you look at a world defined by what's available today, concepts like, the word recovery has been talked about in the context of addiction and serious mental illness. The concept of cure used to be talked about, right? Freud's technique was the talking cure. It wasn't the talking symptom suppression. And so the idea that as we accumulate more evidence, we might have to sort of evolve the taxonomy, the vocabulary we use to describe change in people. The curves that we saw in our study for response, to me, looked like a state change, right? People started in one place, had their session, the next day they are in a different place, and for at least the 12 weeks that we watched them, they stayed in that place. We weren't seeing a tailing off or a degradation of effect. And so, do we talk about, if we start to see effects that last longer? Certainly the concept of sustained remission is a very real thing. But when I think about this more from a sort of a diagnostic, philosophical place, if someone had GAD or MDD, and now they've gone, you tell me how many weeks, months, years without it, well, at some point, I suppose they don't need to still have it. And so, that to me is sort of definitionally a cure. We just haven't been able to talk about cures in psychiatry in quite some time, with the exception of psychotherapy. In psychotherapy, there very much is the ability to induce, not as quickly as, say, a single session in most cases, but there is the ability to induce a state change and a lasting change in how someone feels in the world and feels about the world, feels about themselves. So we're certainly extremely hopeful, and we're doing things in our pivotal studies, in our phase 3 research, that ought to at least be able to demonstrate or assess the duration of efficacy, the single dose, and then we'll be able to look at patterns of redosing as well.
This is it's all just following the space and the work MindMed's doing is it's very exciting, and I can't wait to see more data readouts in the future, especially from MindMed. I think 2026 is going to be a very, very busy year of readouts for the company. So I'd like to move on here to maybe speaking about LSD specifically. You know, a lot of people within the psychedelic medicine space are very excited about psilocybin and DMT. You hear these two psychedelics, I think, spoken about probably the most frequently. You know, when looking at the phase 2b data for MM120 in generalized anxiety disorder, and then looking at the design of the phase 3 programs in GAD and MDD, I couldn't help but notice that the company is looking at only one dose before the 12-week endpoint in these trials. You know, with other companies working on psilocybin and NN-DMT, we see, you know, companies exploring 2 doses before the primary endpoint in their trials. Maybe, and I would like to hear more from you about this. This speaks to the durability of effects that LSD has shown so far in clinical trials. So, you know, after seeing how impressive the phase 2b results in GAD were, I would like to ask, is there something here with LSD, MM120, that people might be missing?
We went with one dose again because one dose showed what it could do in phase 2 and gave us great confidence to move forward with that single dose paradigm. And
Yep.
you know, if you can get that many people better and get a successful trial with a single dose, then that's what we should do, and that's why we're doing it. The extension period in those studies allows for a few things, but maybe most importantly, the extension period, though it does have open label treatment in it, and just for comparison, if we were doing a daily drug, let's say we were developing another SRI, and through the double-blind period, people got, every day they got a dose of drug versus placebo each day. When they enter an extension period, then on day 1 of the extension period, everybody's getting active drug, and they know they're getting active drug. So they become unblinded at that moment of transition. A really neat thing about our paradigm is that people will transition into the extension period, but because it's not a daily drug, because it's an intermittently dosed drug, people stay in the blinded condition until they get until they get an open label treatment. So we are able to look at and of course, everyone stays blinded, right? There's no point at which you unblind the double-blind period after you lock the database, but we don't get unblinded to the initial allocation until then. So people are proceeding in a blinded state through that extension period if and until, like I said, they get a treatment. And so we'll be able to assess the efficacy out until a year from that first dose in a blinded fashion, which is really exciting. And then what we get to see is, with this opportunity for open label treatment, if somebody either loses efficacy, they never had efficacy, they possibly got placebo, which is why they never had efficacy, then they get dosed in this open label fashion, and we'll get to look at what sorts of patterns emerge there for treatment, right? And so other companies, I think, because they wanted to boost their durability, have done these 2-dose paradigms. There's certainly maybe people in that extension period who get dosed, don't have a full response, don't go low enough that they're ineligible, and get a second dose a month later. And so in essence, we'll also be able to, not in a blinded fashion, admittedly, but we'll be able to look at what sort of patterns it takes to capture people and to get their, to get them either into remission or at least substantially lower on the scales.
I see. And I guess, still staying on the topic of LSD specifically, I guess, could you, go over like a little bit about what are the similarities with psilocybin and what are the differences, if you don't mind?
Yeah. So LSD is obviously synthetically derived. It's, it's semi-synthetic, so it starts from naturally occurring precursors. But the nature of the LSD interaction at the 5-HT2A receptor. So I imagine your audience is quite familiar with some of this pharmacology, but I'll just real quickly remind everybody
Thank you.
that so psychedelics, classic psychedelics, let's say, and you mentioned psilocybin, DMT, 5-MeO DMT, mescaline, ayahuasca, which is a combination of essentially it's DMT, but with an MAOI to prevent it from being broken down, and of course LSD. There are acute effects, the things that we conventionally think of as the experience of taking these drugs at high enough doses, which in the case of LSD, remember, we're talking about 100 micrograms, right? So that acute experience is mediated by binding to the 5-HT2A receptor. What that does is a bunch of different stuff, that kind of binding. Now, the fact that we're dosing 100 micrograms, while most of the other classic psychedelics, really all of them, the other ones are being dosed at least a milligram, if not tens of milligrams, and in some cases much more than that, is that we have a very potent drug, which means that it takes a low amount of the drug to highly bind the receptor. There's also what people call a flap or a lid in the receptor that's been implicated in why LSD is so potent and has the effects it does on the receptor. So in essence, the receptor sort of traps the LSD molecule in its binded pocket. Beyond that, for the acute effects, the way we know this, by the way, is if you block 5-HT2A, you block the acute effects of these drugs. Now, the drugs also bind to other receptors in the serotonin system, the dopamine system. So they have multitude of effects in the brain, but that primary acute experience of the drug is mediated through that receptor. And we do see, based on potency and the pharmacodynamics of the drug, we seem to have this tight binding with strong effects per dose, per size of the dose, per amount taken, per exposure in the body. The length of the experience, which obviously over the past few years, people have talked a lot about duration of experience and what's feasible in clinics and what's economic for clinics. We actually think that our 6 to 8 hour experience is kind of optimal for the working day for a clinic. So we think that it provides actually some distinct advantages for clinics that can run one patient, get paid per hour of monitoring for that whole day of monitoring, and one per room, one per bed, one per whatever unit of whatever, whether it's sort of the esketamine model where people are in almost like ER curtained off rooms or separate rooms, but we think that provides some clinically operational advantages. But I'm also struck by the fact that when folks look at mostly in animal models, the duration of what they're calling the critical period that reopens after taking, and this is where we get into the neuroplasticity conversation, but the duration of that opening is longer with LSD than it is with the other molecules you mentioned. Wow. And so whether that's mediated by the length of the experience or something specific about the pharmacodynamics of the drug, we don't know. But in all this conversation, you can't help but sort of get to, well, how do these drugs help people get better, right? The question of mechanism becomes a totally valid one to wonder about.
A quick question about I believe it's, the ODT formulation. Is that what's making the LSD potentially able to fit into 6 to 8 hours in a clinic?
Yeah, let's talk about duration, because people did get sort of hung up on the fact that we were doing a 12-hour minimum monitoring period in phase 2. We agreed to that with FDA. We agreed to that because we didn't have data to make an argument otherwise. And we wanted to make sure we were being as safe and careful with our participants as we could be. We also had that 200 microgram dose group, which can introduce prolonged experience. We were using, in the phase 2, we were using capsules, so powder and capsule formulation. And what we agreed was, okay, well, we'll do a 12-hour fixed monitoring period. We think, by the way, having a minimum fixed monitoring period is pretty important, because if you don't, there are going to be a bunch of people who didn't get any drug who are going to be eligible to leave like an hour or two in. So
I see.
It's a little bit of a double-edged sword, but if you're already worried about unblinding other people on the research staff, and there are a bunch of people who are leaving at hour 1 and a bunch of people staying till hour 8, that creates a pretty point at which folks would say, okay, well, there's more functional blinding. But what we did was we started collecting essentially the symptoms of LSD consumption starting at hour 8. So beginning at hour 8, a study staff member would run through essentially everything that you possibly might feel from LSD and see if people were still feeling it. And we waited for them to leave till they had essentially total symptomatic resolution. So we used very conservative measures. We looked at the curve of those symptoms of drug taking going away. And we then, when we came back for into phase 2, had data to say, actually, for phase 3, why don't we do a fixed 8-hour monitoring period? Again, it's important to keep that fixed period, but we're going to use a checklist and start doing that checklist at hour 5. So we can push back and gather. So what we're doing is effectively gathering evidence for what the appropriate duration of clinical monitoring is. We've also, we think it's really important to use like clinically relevant measures of safety for when people are allowed to leave the clinic. So rather than using like the full everything you might experience from the drug, we've now reduced the things that we're looking at to decide when someone's ready to leave, even though they can't leave till hour 8, but we want to know if they're ready to leave earlier, to things that are relevant to their ability to safely leave the clinic and exist on their own outside of the monitored setting. So we've tried to, in a rigorous way, look at the appropriate duration for clinical monitoring so that when it comes time for us to be out in the world, engaging with payers, engaging certainly with regulators on things like REMS and labeling, that we have a good body of evidence to say, here is what should be happening in the real world, and here are some tools that can make that easier to do in the real world. Because ultimately, of course, the goal is, the clinical research is to prove to our own and to FDA's satisfaction that the drug is safe and effective, and then to have it be able to go into the world and maintain that same safety and efficacy.
Thank you so much for this information. That's very helpful. I didn't know a lot about a lot of this. So I'd like to move on with, MindMed recently announced that they are expanding into major depressive disorder along with generalized anxiety disorder. So first of all, why MDD, major depressive disorder? And you actually did touch on it a bit earlier. So why MDD and not TRD, treatment resistant depression? But you did touch a bit on that.
Yeah. So first of all, you can't talk about GAD and MDD without acknowledging that they are tremendously overlapping diagnoses. The construct, right? These are diagnoses that are defined in the DSM, Diagnostic and Statistical Manual of Psychiatry, that their definitions in the book have about an 80% construct overlap. So you can draw a line between about 80% of the definitions. So unsurprisingly, about 80% of people who qualify for one diagnosis qualify for the other diagnosis as well. There are some differences in acute presentation. GAD tends to be more of a stable state where people are just sort of always at the same. Now, of course, like life events will cause fluctuations. No one's exactly the same amount of anxious one day to the next, but for the most part, you don't think of GAD as being an episodic illness. It's a continuous illness. Major depressive disorder is defined by the fact that it's episodic. So major depressive disorder is defined by having periods of major depressive episodes, which is when someone's depression gets bad, essentially, when they fall into a state of depression. Really interestingly, what we often see is that people with MDD, between their episodes of depression, can be highly anxious. But because they've had a major depressive episode, they get the major depressive disorder diagnosis, because a lot of the attention of the industry has been on major depressive disorder for the last 30 years. There's some reasons for that have to do with the qualities of SRIs and what they're best able to treat. But what we saw was that there's this huge unmet need in GAD. And when you think about what it actually means to have a resistant depression, often it means that the residual symptoms are pretty anxiety-looking symptoms. So that even when people are partially in remission from major depressive disorder, their symptom burden can be largely anxious. Now, because of that overlap, and because of the remarkable effect that we saw on the depression scores for people in the phase 2, so these were patients with primary GAD, but they were allowed to have an MDD diagnosis. They
Okay.
just couldn't be in a major depressive episode. And what we saw was this remarkable, effectively the same change in the MADRS, which is the depression scale, as we saw in the HAM-A, the anxiety scale. So that gave us a lot of confidence to now go into a population of people who are in a major depressive episode, in addition to our GAD patients. And the hope here is ultimately that what that does is it makes us the go-to drug. If we get approved for both these indications, they're both in the label, it almost eliminates the need to ask the question, oh, is this MDD or GAD or combination? Because we cover that whole waterfront.
That's awesome. So, MindMed is also working on, R-MDMA. Some people might not be aware of this. So, what is the path forward for this program currently? What stage of development is it in, and what are you looking at it to treat?
Yeah. So remember earlier when I said MDMA doesn't work unless you get therapy? In our case, we've got a really interesting paradigm with R-MDMA. So for folks who aren't familiar with it, MDMA that's been used by others is a racemic mixture. So it's got molecules that are left-handed and right-handed. And everybody learned this at some point in high school chemistry. And if you don't interact with it on a daily basis, you can be forgiven for forgetting it. But molecules that are chiral, that means that they can be non-superimposable mirror images of themselves. So literally think like your hands, right? Your hands can't be superimposed over one another, they're mirror images. And in the case of MDMA, the right enantiomer, and this is just a totally interesting thing about neuroscience and the way that our brains work, which is that the two enantiomers of a molecule can behave really differently from one another on receptors in the brain. Once you think about it for a minute, okay, that makes sense that they do different things. And in the case of MDMA, the right enantiomer seems to be the more serotonergic enantiomer, so the one that drives the more classic MDMA feelings of closeness and openness, communication, awareness of your own feelings. The S enantiomer, the left enantiomer, seems to be more amphetamine-like and more dopaminergic. So it drives sort of more of the get up and go feeling that the racemic mixture does both of these, of course. And our hope is that unlike the other development paradigms people are using for MDMA, that our R-MDMA product is not a psychotherapy enhancer, but rather we're thinking of it as a daily drug for folks with ASD, autism spectrum disorder. And it's neat in that regard because the direct effects of the drug, promoting feelings of closeness, awareness of one's own feelings, awareness of other feelings, correspond with the things that bother folks with ASD, in some cases, not all cases, about being in the world and that folks struggle with and wish could be made better. So in a world where ASD drugs are largely to date about either treating comorbid disorders or about behavioral suppression, our hope is that this would be an enabling drug for the core symptoms of ASD, and that it doesn't go with therapy. It's a daily drug. People take it if it helps them, and if it helps them with certain activities in their life or with certain interactions, that's great. The model is very much analogous to psychostimulants like Ritalin and Adderall in the treatment of ADHD, right? People take it while the drug's on board, they're better able to do school or work or life or whatever, and then the drug goes off, and it doesn't induce, unlike MM120, the attempt is not here to induce lasting change once the drug is gone, but just to enable someone to function in a way that's more preferable to them while the drug is on board.
I see. And so could R-MDMA chronic dosing potentially help with maybe my understanding is people on the autism spectrum may struggle a bit with social anxiety. Could it help address some of those symptoms?
We certainly hope so, and we will test for it. So what we're doing with this drug, this is a very different stage of drug development. It's a higher risk stage of drug development. We know less about the drug. We are working through safety studies because we obviously this is starting from the beginning. We have to do safety studies. Our goal here is to take that to figure out the right dose, to figure out the patient population we want to look at, and to figure out a battery of assessments that we can use to look for drug effect. The goal here is to get evidence of drug effect in a new drug in a population of people who, who are quite diverse in what their experience of the world is, their experience of their lives, their experience of the disorder even. I mean, obviously there's debate in the community about whether it's even to be called a disorder in certain contexts. So the trick here is for us to get evidence of efficacy that gives us confidence to bring the drug forward.
I see. Thank you for the overview. I'm looking forward to also following the progress with this program as well. It's MM402, correct?
That's right, MM402.
So, to wrap things up, Dr. Karlin, and thank you again so much for coming on the channel. I've really enjoyed this discussion. What are you looking forward to for the next months and years ahead? What should people be looking out for when it comes to MindMed?
Yeah, I mean, we are right here at the beginning of two pivotal programs, right? And so sitting here today with you, we are doing all of the things that it takes to get large pivotal programs off the ground. We have a phenomenal clinical ops team, folks who care really deeply about these development programs, who are deeply invested in the success of the programs and in running high integrity, high fidelity studies. We obviously worked with a lot of the sites who operate in this space and are close with investigators, close with those sites. So right now it's really about organizing all of the various resources needed to launch these studies and to make sure that we're running the best studies we possibly can. The drug has all of the historical evidence it has the evidence we've gathered from our phase 2. We've talked a little bit about some bioequivalence work we've done and other clinical pharm studies we've done. When you have a drug with this much history and this much promise, I think we all just feel this, it's incumbent on us to do this as best we possibly can, so that our eyes are looking forward. And you mentioned 2026. We're looking forward to getting data that are as meaningful and as representative of what actually happened in the trial as possible. And then, of course, looking forward to being able to bring this entire package that we've worked to create and are working to create to FDA, so that we can have those conversations from a whole different. We started with you asking about how are things different. They're different because from the outset we've been thinking about how to put together a package that will be familiar to FDA, that will look the way they're used to seeing successful filings filed, and that it will give us everything we need to work with them to get to a label, and ultimately, if we're able to get there, to get out into the clinical world.
Very exciting. I'm really looking forward to following the progress. Dr. Karlin, thank you so much for coming on the channel today.
Yeah, thanks for having me. This was really fun.
Thank you.